The storage stability of extracellular vesicles is an important parameter for their prospective therapeutic use. Our protocol provides a readily applicable tool for evaluating how storage affects the vesicles. The main advantage of our technique is that, by introducing glucuronidase as an exogenous marker, extracellular vesicles derived from different parent cells can be easily compared regarding their storability.
Asymmetric flow field flow fractionation, or AF4, is a mild alternative to size exclusion chromatography purification for very sensitive compounds, because the compounds encounter less shear stress in the channel.
As glucuronidase is a sensitive enzyme, the quality of your results benefits from thorough planning and carrying out the steps for purification and analysis back to back.
After culturing the cell line of interest according to standard conditions for those cells, replace the supernatant with serum-free or extracellular vesicle depleted medium, and return the cells to the cell culture incubator for an additional 24 to 72 hours. At the end of the incubation, collect the supernatant from each flask and centrifuge the samples to sediment the cells. Carefully collect the cell-conditioned medium without disturbing the pellet, and centrifuge the medium again to remove cell debris and large aggregates. Then carefully transfer the supernatants into ultracentrifuge tubes. If using a fixed-angle rotor, mark the orientation of the tubes in the centrifuge to facilitate